Okay, so I think I mentioned in a previous lecture that after our discussion of carbohydrates, we'd be moving into proteins. But looking through the textbook, I realized that the section of the textbook following carbohydrates is actually lipids, not proteins. And I know I'm not always following the textbook sequence to the letter, but hey, we'll try something that is a little bit out of the sequence that I normally would go through the content. Part of the reason why it would make sense to me to go into proteins next is because proteins and polysaccharides are similar in the sense they're made up of long chains of monomers, right? A polysaccharide like starch or like glycerin is made up of a long chain of monosaccharide, monosaccharide, monosaccharide. It could be hundreds, maybe even 1,000 monosaccharides that get put together to form a single molecule of a polysaccharide. It could be a few hundred, maybe even 1,000 amino acids that get together to form a long chain of a polypeptide, which is the foundation of protein structure, right? Lipids don't have that, okay? So lipids technically aren't really a polymer, although, as I said in a lecture a few times ago, that at least in the case of the triglycerides, you have 3 fatty acids that come together into a cluster of 3. They're hooked together by glycerol, and we'll be seeing that in our discussion. And I think the thing to point out here is that all lipids are not structurally related to each other, even that. I mean, we have those glycerol-based molecules, but we also have a whole group of molecules that are based on steroids, on the steroid structure. Cholesterol, all the steroid hormones, they've all got a structure that is distinct and very different from that theme of fatty acids attaching to a glycerol. And we'll be getting to that with today's discussion. Okay? What unites all lipids is the fact that they are largely hydrocarbon, which means that they're going to be nonpolar with their covalent bonds and hydrophobic, right? Now, they're not entirely hydrophobic all the time. There's going to be something that makes them biologically active, but their overall structure is hydrophobic, which tends to make them greasy. They tend to be non-water soluble, or at least partially non-water soluble. That contributes to some of their functional significances, right? Now, lipids are important for a few reasons. We think of them as having structural function, in particular with membranes. Membranes of cells are always going to be made of phospholipids. Okay? Animal cells are also going to have cholesterol in them. And so the structure of what keeps an animal cell together is the membrane, and what keeps the membrane together are the interactions between phospholipids and the water on both sides of the membrane. Okay. Now, the other thing that lipids are important for is with energy storage. We think of fat as an important energy storage molecule, and the triglycerides are kind of responsible for that. Okay. And there are a few other functionalities that may come into play at some point in our discussion. Hormones, we've actually talked about those already. I showed you a picture early from a textbook that showed a male line and the structure of cholesterol, a female line and the structure of estradiol. We also think of fats as having some importance in terms of body structure. Fats could be considered, yeah, I know it's kind of funny to think about cushioning. And yeah, having a little bit of extra adipose tissue on your backside gives you some degree of cushioning, but that's not really what I'm thinking about, right? Your kidney is a very delicate structure, and it's got shock absorbers all around it, right? If you've ever actually seen a kidney, there's a lot of fatty tissue around it. And the general idea is that fatty tissue is protecting the kidney from the jolts that the kidney is otherwise subject to, right? And so you could think of there being an important kidney function, cushioning function of fats, even for the skinniest person, in terms of their ability to protect organs like the kidneys, right? And insulation, obviously. That's a layer that a really skinny person would not be having compared to somebody with a more significant proportion of body fat, okay? So let's go through a couple of points here. I think this is a review for us. The idea that we have a glycerol. Glycerol is this molecule, C, C, C. I think I drew it for you earlier, and I showed you that it was a triple alcohol. You got an OH there, an OH there, an OH there. Okay, and you have hydrogens in all the other spots for the carbon. So the bonds are going to be filled by hydrogens, and so you actually have 3 carbons, 3 oxygens, and 8 hydrogens. Right? So that's glycerol. And basically, I said earlier, I said that you can attach a fatty acid there, you can attach a fatty acid there, you can attach a fatty acid there. If you do that, you've got a triglyceride, right? On the other hand, if you put a fatty acid here, a fatty acid here, and in the third spot you put a phosphate group, okay, then you would have a phospholipid. Okay? The two structures of triglycerides and phospholipids are actually pretty similar to each other. And again, we're going to be reiterating this. A little bit of repetition is not necessarily a bad thing. Okay? And we'll stop there. We'll pause there, look at some pictures in the textbook, maybe do some drawings, and come back to this this slide share. Okay, so next share, we're going to go to the textbook. This is chapter 3, section 8. It talks about things we've talked about before. Here's the molecule that I showed you, glycerol. There's a fatty acid. Again, if you're not doing this, you should, right? You should say, okay, well, it's a fatty acid because it's got this carboxyl group right here. Carboxyl group makes it an acid. It's fatty or greasy because this long area over here is nonpolar. Right? And so with a dehydration synthesis reaction, we're taking a water molecule out, and we're going to be attaching, we're creating a covalent bond between this carbon and that oxygen. Right? And that's how we attach a fatty acid to a glycerol. All right, and so I know we've done that before, and so we don't need to reiterate. Clear my drawings, move over to the mouse, okay. And this is what we have for a triglyceride. Here's glycerol at the top. Here are the 3 fatty acids. Now, a point here that I need to take advantage of is the idea that we've got a little bend in one fatty acid. Okay, the bend occurs where you've got a double bond in the chain of carbons. Okay, these other chains, the first 2 chains have no double bonds, at least no double bonds between the carbons. Obviously, there's a double bond between the carbon and the oxygen. That's not the one that we count. If you have no double bonds, here's a good word for you. We say the fatty acid is saturated. Okay. You've heard of saturated fats. Saturated fats are the triglycerols, triglycerides that have only saturated fatty acids, right? If you have a double bond, at least one double bond, you are a fatty acid that is unsaturated. And I know that's kind of like what's coming up next on my outline, but I couldn't help but seize this opportunity to identify that for us. Okay. I think if I scroll up, it even takes me to a phospholipid, right? So types of fats. Oh, and by the way, saturated fats tend to be more solid. Unsaturated fats tend to be more liquid. So if you think about the difference between a solid fat like butter or lard or coconut oil, coconut fat versus a liquid fat, we're talking about the state at room temperature, right? Okay, so if you're in a 20 degrees Celsius room, is the fat going to take a solid form or is it going to take a liquid form? Well, that has largely to do with the degree of saturation in the fatty acids. The more unsaturated fats you have, the more unsaturated fatty acids you have in your structure, the lower the melting point, right? Lower melting point means that you're going to be more likely to be a liquid at room temperature, right? So if the melting point is 5 degrees Celsius, right? If you take olive oil, put it into a very cold refrigerator, it will turn into a fat, a solid fat, right? However, if you leave it at room temperature, it's going to become a liquid, right? Coconut oil, on the other hand, if you have it at room temperature, 70 degrees Celsius, it would be kind of pretty much a solid, okay? If the temperature got cold, right, in my house during the winter, the coconut oil stays a solid pretty much the whole winter. However, when it gets summertime and you have hot days and the kitchen stays hot, the coconut oil turns into a liquid and I can pour it, right? So yeah, that's the case for both plant fats, like the difference between olive oil and coconut oil. Coconut oil has a much higher degree of saturation, absence of double bonds, whereas olive oil, which has an abundance of monounsaturated fats, fatty acids, tends to be a liquid, right? Now, remember, olive oil will solidify in the refrigerator because it's got a degree of saturation that's kind of intermediate. If you have a super unsaturated or a polyunsaturated fatty acid, and that's composing the majority of your fatty acids, then you stay liquid even though you're in the refrigerator, right? So like canola oil would be a good example of a liquid, a very liquid oil, okay? It will eventually become solid. If you put it in the freezer, you could turn canola oil into a solid, but it's not going to happen at normal temperatures, even in the refrigerator. Okay. So I think that is covering everything that we needed to from that chapter reading. The next section is, let's go back to the other share whiteboard, because that chapter, that little chapter on trans fats, I appreciate the public health value of explaining that, yes, saturated fats were historically identified as bad. Unsaturated fats were better, and if you catalyze, if you apply a platinum catalyst to liquid oils like canola oil, you could actually turn it into a solid fat with the same unsaturated fats that you had before. The difference, though, that they never really get into is what makes a trans fat as opposed to a cis fat, right? So let's look at this again. We've talked about fatty acids and their structure. What we wanted to identify the variation, and certainly we could have variation in the length of that hydrocarbon bond, that hydrocarbon chain. And we've also said that unsaturation refers to double bonds in the structure, right? So saturated fatty acids have no double bonds between the carbons and the hydrocarbon chain. Unsaturated fatty acids have at least one. The more saturation you have, the more unsaturation you have, the more liquidy you are. The melting point goes down as you add more double bonds. And yet the exception to that is when the double bond happens to be trans, because you could have a trans fatty acid and that would tend to stay relatively solid. Okay? And so this is a very neat thing to think about, because when we draw hydrocarbon chains, remember that we're kind of used to thinking of a hydrocarbon chain as being C, 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 C. And we know that maybe the carboxyl group is over here. That's the thing that makes it an acid. Then we've got, I'll make this a very short one, right? H, 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 H. So fill in the H's. And we know that we can depict this by saying, okay, well, this carbon is going to be represented by this point. And so all we have to do is to identify a double bond to an oxygen, an O and an H there, right? This carbon is going to be this point in this zigzaggy thing. And so I've got 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6, right? And so this molecule, as I've drawn it, okay, get rid of these little these dotted lines. These two molecules, these two pictures represent exactly the same molecule. It'd be a fatty acid with a 5 carbon chain after the carboxyl group, okay? Now, what happens when you have a double bond, right? So let's say we're going to be changing the structure so that instead of having all the hydrogens here, okay, we're going to be putting double bond between those two carbons, right? Now, when that happens, that would be like, let's see, 1, 2. That would mean that this bond would become a double bond, right? Now, when that happens, this last bond right there, it could either come in the up direction or it could be coming in the down direction, right? And so there are really two ways for us to draw this new molecule. It would still have 1, 2, 3, 4, and that's double bond. Here's the O, OH. Okay, now we could have going in the up direction like this, or it could be coming in the down direction like this, right? And so there are two forms for the unsaturated fatty acid, even though the saturated fatty acid would have a straight chain, right? Okay, so this is trans version. This one with a crook is the cis. Cis is crooked, trans is straight. Got that? And so if you think about it, these are the ones that are going to make the fatty acid, the triglycerides, more liquidy. Okay, a trans fatty acid is going to stay relatively straight. This will allow it to be more solid. Okay, so think oil versus margarine. Okay. Well, margarine is made from an oil like this, which they've rearranged the fatty acids so that instead of being a cis fatty acid, it will be a trans fatty acid, which will make it nice and solid. And these trans fatty acids are the ones that are kind of being given credit for a lot of human health issues, right? So the trans fatty acids, the ones that have been modified from their natural cis state, are the ones that are being given credit for a lot of heart disease. Okay, so yeah, there's that neat little section in the textbook where it talks about trans fatty acids in the diet, although it never really bothers to tell you what a trans fatty acid is. And that kind of bugged me, so I needed to fill that gap. So here we are back on the trans fatty acids studies. And so it talks about the relative risks of heart disease. Then this is kind of like down here is the common wisdom. We know that diets rich in animal fats like beef are going to be having a lot of saturated fats in them. Saturated fats contribute to the formation of those bad LDL, the bad cholesterol, low density lipoproteins. And the general idea is that you don't get those plaque forming low density lipoproteins in your arteries if you primarily eat a greater abundance of these unsaturated fats. Okay. Now trans fats are unsaturated, but they're not good for you because they're actually doing, making it even more likely for you to develop heart disease than even the saturated fats like lard, okay? Anyways, I think that looking at this chart, It's a good chart that kind of helps you develop your ability to you think about how we want to depict things graphically. Right? I think from that standpoint, this chapter is really nice. You get a nice visual image that tells you, okay, here is what a trans fat does in terms of your risk for developing heart disease. Okay, and that's what you want your diagrams. If you ever present a figure to me in your lab work, you want to make sure that it is actually telling me something and giving me a nice visual that emphasizes a point that you want to be making, right? So it's good from that respect, but from the standpoint of our understanding of the structure of fatty acids and the structure of lipids, it misses out on that opportunity to address the difference between cis and trans. Okay, so anytime that you see a crook in a fatty acid, so in other words, in all the fatty acids that we talk about in our class, these crooked fatty acids you see like that, that means that it's a cis fatty acid. A trans fatty acid would simply be a straight line, or it would stay straight. Okay. That's not the point of what's in this particular diagram, though. Okay, I think the point of this diagram is that we're looking now at a phospholipid. Phospholipids, remember we've got the two fatty acids, and the third position, we've actually flipped that around so that the phosphate head is pointing in the up direction. There's also this other thing on top of it, it's the choline molecule, also attaching to the phosphate head. Don't worry about that so much. I do want you to look at the fact that the phosphate head is charged. Okay. So we've got this structure here. In contrast to the rest of the molecule, it's got a charge. And it's going to make it hydrophilic. Okay. The rest of the molecule, everything from that point downwards, is going to be completely nonpolar and hydrophobic. Okay. And I know you're not used to looking at molecular structures and making determinations from that. I know that BIO110 students tend not to be terribly practiced in doing that, but that's the whole point of what we're doing with these lessons. Look at a molecule, use the information that you've just gained about covalent bonds and water, and tell me why it is that the water molecules do not want to interact with anything down here, but the water molecules do want to interact with this charged head that we have on the top of the phosphate. All right. So this, we sometimes would call the head, phosphate head, tail. It's mostly hydrocarbon. Okay. And so we could actually look at this molecule and say that this structure that we're looking at here, the head and the tails, is the representation of the molecule that we have over on the left. This is the part that will be hydrophilic. Okay. Water is happy to interact with the phosphate heads, but the water does not want to interact with the hydrocarbon tails. This will be the mostly water-free, greasy middle layer. Of a membrane. Okay. And what we're looking at with this structure is like a whole bunch of phosphates forming a layer, one layer. And it's not just a random layer. The phosphate heads are all lined up on the same side of the layer. They're up against the water. The nonpolar tails of the phospholipids are in the opposite direction. You've got another layer underneath. This is layer number 2. And the second layer, phosphate heads are all arranged so they're pointing downwards and are interacting with the water molecules down here, okay? And the nonpolar tails are pointed upwards and are interacting with the nonpolar tails. In other words, all the nonpolar molecules are sticking together to form this kind of greasy middle layer of the membrane, and all the water molecule, all the phosphate heads are pointing to the outside and interacting happily with the water molecules. This is a structure that forms spontaneously if you mix phospholipids with water. You get these sheets forming because this is a natural state, a natural way that the phospholipids will want to interact with water. Okay. And so the other thing about this is that the membrane, if this is the basis of a membrane, there's nothing that really holds the membrane as a solid. The membrane is actually kind of like a two-dimensional liquid because you've got a layer of phospholipids. Each individual molecule is not covalently bonded to anything that's nearby. Basically, every phospholipid is kind of freely moving around. They can shift around relative to each other as long as they're staying with their hydrophilic heads pointing to the water side and hydrophobic tails pointing to the middle greasy layer of the membrane side. Each molecule is free to move around as it wants to. Right? They can move laterally in all different directions. Right? The same goes to the bottom layer. And so sometimes we often refer to the basic structure, the basic fabric of biological membranes as being a phospholipid bilayer. Phospholipid bilayer or a bilipid membrane, right? Membrane just means it's a division, right? So it could be lots of things. Okay? In this particular case, we're talking about the membrane that is involved in cell membranes, plasma membranes, internal membranes like the nuclear membrane, the membranes of the organelles like the mitochondria and the endoplasmic reticulum. All of those cellular membranes are based on a phospholipid bilayer together with a fair amount of protein. Okay, and so that's the significance of our phospholipid. And I think that we are moving on to the next picture, which is that of cholesterol, right? And so cholesterol is at the bottom of this. Let's see, this chapter. Cholesterol is relatively easily recognizable because, well, it's got 4 rings. Okay, we call them the A, B, C, and D ring. For me, it kind of helps that the D ring is a 5-membered ring. 6-membered rings are far more common in molecular structures. Okay, 5-membered rings are somewhat rarer, and the 4 rings for the D ring is a 5-membered ring. D has 5 edges. Okay. This is a formula that I use to recognize a molecule that is a sterol or steroid. Okay. Now, in the case of cholesterol, we look at the structure. Okay, again, this is practice for you. We only note one point at which we have something other than hydrogen and carbon. Okay, this is the one point of hydrophilicity. What is hydrophilicity? Well, hydrophilicness, if you like. Okay, we've got the OH, that's the hydroxyl group, which basically makes cholesterol an alcohol, right? In fact, all of the N is an indication that it is a alcohol, right? Okay. Now, the rest of the molecule is entirely nonpolar. So kind of similar to what we have in a phospholipid is that we've got a polar part and a nonpolar part. And what happens when cholesterols are interacting with phospholipids is that the hydroxyl portion is going to line up with the phosphate head. Okay, and the nonpolar parts are going to interact with the hydrocarbon tails, right? And so the structure of cholesterol is going to be contributing to that phospholipid bilayer we were looking at before, and it's going to make the phospholipid bilayer somewhat more stable, at least in the case of animal cell membranes. Other properties as well. We talk about membranes quite a bit in an upcoming module, right? Questions? Yeah, the last section of the chapter is just a little bit of a public health blurb about you should not use anabolic steroids if you're trying to bulk up. Don't do it, they're dangerous. Although one thing about steroids is I don't think that they have a picture of the actual molecule, but if you have this molecule in your mind, if you have a mental image of what this molecule looks like, if you could actually see an anabolic steroid, okay, that's not it. And so there's no actual picture of a steroid here, but I think if we back up, yeah, if we back up and look at chapter 3, section 2, now revisit testosterone. Remember this picture? Estradiol, male hormone on top, female hormone below. Are you seeing what I'm saying? Right? 4 rings. The 4th ring, the D ring, has got 5 members. That's what makes these kinds of molecules easy to recognize as being steroids. Right? So a lot of hormones are steroids, and that's something for you to be aware of. And not all hormones are steroids. There are a lot of hormones that have different structures, but steroid hormones are among the some of the more common ones. Okay. Now, going back to our whiteboard, at this point, we're just checking to see if I've covered all of my bases. Double bonds may be cis or trans. Phospholipids are, well, I don't think I've ever used this word in our discussion. Probably not even that important, but ambi means both ways. Valent means attaching to. So basically it's got the 2 sides to it. You've got the hydrophobic part and the hydrophilic part, okay? Which part of a phospholipid is hydrophilic? Is it the head or is it the tails? Yeah, okay, well, I'll let you review that. Phospholipid interaction with water is what causes it to form a membrane. Okay? And we talked about how steroids are 4 ring structures. That D ring is a 5-membered ring. It can be used to stabilize membranes. Discussed that briefly. And we also said that they're related to steroid hormones. And I showed you the picture with the lions to validate that. Okay, so that's it for this lecture. I will talk to you next time about proteins.